Hello everyone. So this is the first time we meet. First I would like to introduce myself. My name is Alžběta Blanková and I work as an assistant at the Department of Histology and Embryology of the Second Faculty of Medicine. And also I work as a medical doctor at the Department of Forensic Medicine and Toxicology in the Regional Hospital Liberec. And I work as a coroner service in the capital city, Prague. So today we should discuss the histology of nerve tissue, and we will talk about the basic characteristics of the nerve tissue and about the cells we can find there and their differences, their functions, and so on. So let's start with that. So what is the basic characteristic of nerve tissue? Nerve tissue is specialized for input, processing, and transmission of information, which enables nerve tissue to coordinate the majority of functions in organism. So the nervous system, which is composed by nerve tissue, enables the body to respond to the changes in its external and internal environment. Generally, the nervous system controls and integrates functional activities of organ systems in the body. What can we find within the nerve tissue? We can find a lot of cells with long processes, and they are connected together to create a very difficult network. And between those cells, there is only a small amount of intercellular matrix. Those cells we can find in the nerve tissue are divided into two groups. So first group are excitable neurons or neural cells, which are the functional units of the nervous system. Nerve cells are specialized to receive stimuli from other cells and to conduct electrical impulses to other parts of the system via their processes. The other group are the supportive glial cells. These supporting cells are non-conducting cells, and they are responsible for maintaining the proper environment for neurons. Yeah. Okay. Nerve tissue creates nervous system. Nervous system can be divided anatomically into the central nervous system, which is called CNS, which consists of the brain and the spinal cord, located within the cranial cavity, spinal canal respectively, and the peripheral nervous system called PNS, which consists of the cranial, spinal, and peripheral nerves, and their ganglia, of course. Ganglia are accumulations or collections of nerve cell bodies outside the CNS. In the CNS, for example, in brain, we can see two types of matter which have different coloring. The first is gray matter. Gray matter contains neural bodies and short unmyelinated processes. And white matter, the color, whitish color, is caused by myelin content. Myelin is a lipoprotein complex covering individual axons to make the conduction much faster than in the case of unmyelinated processes. So we'll talk about this special myelin later. This is just the image of organization of those cell types within the CNS and PNS, just for illustration that that's really, really difficult network. So let's go back to those two types of cells. Neurons. I've already mentioned that those are excitable cells capable of information transmission. That means they conduct the signal or electric impulses to other parts of the system. Glial cells, which are called neuroglia sometimes, they support, they protect, and they nourish neurons. So their purpose is to ensure a proper environment for neuronal function. There are four types of glial cells: oligodendrocytes, astrocytes, microglia, and ependymal cells. In this case, we are talking about central nervous system. In the peripheral nervous system, there are different glial cells. We will talk about it later. So we can divide that into the central glia and peripheral glia. These are the central glia. So neurons, as I mentioned, are the basic functional units of nervous system. They are responsible for input processing and transmission of signal. They conduct the electrical impulses and create a very difficult network. They consist usually from a cell body, which is sometimes called soma or perikaryon, and a lot of processes, shorter processes, which conduct the impulses toward the cell body, are called dendrites. And the other type of process, which conducts the signals from the cell body to the periphery, is called axon. So the dendrite and axon. Neurons are very variable cells. They vary in size. The smallest are cerebellar granular cells in the cerebellum. They are about 5 micrometers in diameter. And the biggest are the same as in the cerebellum, cerebellar Purkinje cells. And they measure about 150 micrometers in diameter. So those are maybe the biggest cells of a male body, just male, because biggest cell of the female body is the oocyte, I think. So this is the general structure of typical neuron. This type of neuron has a lot of processes. You can see shorter processes called dendrites. These dendrites, they transmit, they are like receptor processes. They receive the stimuli from other neurons or from the sensory organs from the external environment. And they receive the information from other neurons or the external environment, and they carry that information to the cell body. This direction is called centripetal. Centripetal. So they conduct the signal toward the neural body. They are shorter usually than the axon, and you can see the dendritic tree, which is like branching of the distal portions of dendrites with the widened endings called dendritic spines. These are synapses between the neighborhood neurons and this neuron. Then you can see neural body, which is like the metabolic center of the cell. It contains the nucleus and the nucleolus, usually one, and a lot of rough endoplasmatic reticulum and Golgi apparatus, a lot of mitochondria and other substances, including the microtubules and ribosomes and so on. The neurons are the typical protein-producing cells. That means that they have a lot of rough endoplasmatic reticulum and prominent Golgi apparatus. Then you can see another process, which is much longer than these dendrites. This process is called the axon. Axon conducts the signal from the body to the periphery. The periphery, this could be another neuron like that, you can see here, or it could be an effective organ, like, for example, a striated muscle, which is innervated by this neuron, and its movement is caused by the nerve stimuli from this neuron. So this axon starts here with a part called axon hillock, and then it goes to the periphery to the another neuron or the effective organ, where it ends with synaptic ending. Synaptic ending or synapses is a specific communication and connection between neural cells, and it consists of presynaptic part, which is usually the axonal ending here. So that's the presynaptic membrane, this one, in axonal ending. Then there is a small intercellular space called synaptic cleft, and the other part, postsynaptic membrane, which is usually part of another neuron here. For example, it's dendrite, which is receiving the information from this presynaptic neuron. But it can also be an effector organ, as I said. You can see that the axon is longer than shorter dendrites, and it is enwrapped by myelin sheath. That's this blue part. Myelin sheath is not continuous. These segments, it's segmental, these segments of myelin sheath have between them parts where the axolemma, which is like cytoplasm membrane of axon, it lacks myelin sheath. That means that this part is bald. It's not enwrapped by myelin. And these junctions between the myelinated segments are called the nodes of Ranvier. Myelinated parts are electrically isolated. That means that the electrical current is not conducted here. And this causes that the action potential, which is initiated here, it has to jump from one node of Ranvier to another, like that. This conduction is much faster than continuous leading, and it's called the saltatory conduction. The three basic types of neurons, according to their morphology, are the multipolar, bipolar, and pseudo-unipolar neurons. First group, multipolar neurons, are the most numerous in nervous system. They have more than two processes. It means that usually one is dendrite and one axon. It has more than two. It means usually there is more dendrites than axons, but there are also some neurons which can have more axons. This is normally the typical example of multipolar neuron. It means that it has a lot of dendrites, for example 200 dendrites, which ensures the communication with the surrounding neurons, and one longer axon which leads to the periphery to communicate with another neuron, with synapses, or with effectory organ like a striated muscle or gland, for example. So this is multipolar neuron. The other, bipolar neuron, has only two processes. That means one axon and one dendrite. These are found, for example, in retina, bipolar cells of retina, and in the cochlear and vestibular nuclei, and in olfactory mucosa. So we can say that we can find them in some sensory organs. The last type are the pseudo-unipolar neurons. They have only one process, which branches shortly after leaving the perikaryon into one dendrite and one axon. This causes the T letter shape of this neuron. We can find these neurons in sensory ganglia of spinal and cranial nerves. So this is the most common typical pseudo-unipolar neuron, which has dendrites. You can see that dendrites are usually unmyelinated, and they conduct the stimuli toward the neural body. This direction is called centripetal, and one axon which conduct axon potential from the nerve body to the periphery, which is called centrifugally. So this is the multipolar neuron. This is bipolar neuron with one axon, one dendrite. Usually the axon is not myelinated as well as the dendrite. And the third, last type, pseudo-unipolar neuron, has the body and very short process, which is shortly after leaving the perikaryon, branching into dendrite and axon. Dendrite receive the stimuli from the periphery, and axon leads or conducts the signal toward the periphery. So that was the morphological division according the shape of the neural cells and number of processes, and this is the functional division. First are motor neurons, which are like efferent neurons controlling effector organs. Second group are the sensory neurons. They receive the information from the body and from the external and internal environment, so they conduct the signal to the center from the periphery. And the third type are interneurons, which are short neurons which form complex functional connections between the sensory and motor neurons. So three types of neurons. Similarly, we can divide neurons according their function into projection neurons, which are usually the motoric one, or sometimes called neurons type 1, which have the big perikaryon, long axon, and they conduct impulses on longer distances. And interneurons, which are represented by neurons type 2, those are Golgi type 2. They are much smaller, they have short axons, and their axons do not leave the area where the neuron is situated usually. So they are pretty short, and they ensure the communication between those big projection neurons. So what about the parts of neural cell, nerve cell? First, the perikaryon, or the cell body, contains nucleus, which is usually big, rounded, and achromatic, with prominent nucleolus. Of course, that this is the sign of great transcriptional activity. The nerve cell is the typical protein-producing cell, so that's typical for that. Rough endoplasmatic reticulum with a lot of polaribosomes on the surface together form the accumulation or the collections called the Nissl bodies. Nissl bodies. They can be stained, for example, with crystal violet. So Nissl bodies are accumulation of polaribosomes within the perikaryon. Then, of course, Golgi apparatus in perikaryon exclusively. It produces the transport and secretory vesicles, and of course, a lot of mitochondria. Mitochondria are gathered mainly in axonal endings and in perikaryon. And of course, the perikaryon is metabolic center of the cell, and it ensures nutrition for processes. For example, for axon. Axon does not have the proper metabolic function. It is dependent on the perikaryon. And so the nutrition has to be transported into the axon by cytoskeletal structures serving as a railway. This railway is made by microtubules, and of course there are neurofilaments which can be stained or impregnated by silver or gold. And the axonal transport is ensured by motor proteins like adenine and kinesin, and they use microtubules as a railway, as a rail for that transport. So this is a picture of the perikaryon. You can see the great nucleus with nucleolus. Usually the nucleus is in the center of the perikaryon. Then you can see Nissl bodies, which is like accumulation of rough endoplasmatic reticulum and polaribosomes, mitochondria, and so on. This is a crystal violet with staining of Nissl bodies. And this is a electrogram of the perikaryon. You can see the nucleus with nucleolus, a lot of mitochondria within the cytoplasm, like that, mitochondria, and those are myelinated and sometimes unmyelinated axons, which are nearby the neural body. Dendrites are short processes which increase the surface of perikaryon. One neuron receives information from several neurons, usually from the neighborhood, but it it's not always. Dendrites are ended by dendritic spines. Dendritic spines are endings of dendritic tree, and these are sites of synaptic communication with another surrounding neurons. So this is the site where this neuron receives the information from the other neurons. A dendritic cytoplasm is much similar to perikaryon, but it doesn't contain Golgi apparatus in distal parts. Axon, on the other side, is quite different. The beginning of axon is called the axonal hillock. It doesn't contain any ribosomes and rough endoplasmatic reticulum. And in this part, in axonal hillock, the axon is not surrounded by the myelin sheet, not yet. Myelinated axons have the initial segment where the action potential originates. Initial segment is the part between the axonal hillock and the beginning of the myelin sheath. In this part, the axolemma is rich in sodium channels, which ensures the change of the membrane potential, which is essential for creation of action potential. Terminal part of axon is usually branched. Each branch terminates with specific synaptic ending, and this arrangement is called bouton terminal. It can be sometimes the synaptic endings can also occur along the axon, which are called the bouton en passage, like the buttons in passing. Yeah. So axon, it does not contain rough endoplasmatic reticulum, and as I mentioned, it's dependent on protein supply from the cell body. As I mentioned as well, the nutrition is transported by axonal transport. Microtubules serve as rails for motor proteins like dynein or kinesin, which transport vesicles to the axon, and of course also backwards to the neural body. So the axonal transport is anterograde from perikaryon to the axon and retrograde back to perikaryon. It can be slow or fast. The fastest is about 40 centimeters per day. It's pretty fast. So this is the perikaryon cell body. This is the axon ending with synapses, and this is the axon. You can see microtubules as a rails for motor protein, like that, and they carry vesicles from the perikaryon to the axonal ending, because the neurotransmitters which are released from the nerve ending, they are of course produced in the cell body, so they have to be transported to the synaptic endings, and of course there has to be retrograde transport for degradation of the waste products and so on. So the membrane potential, the action potential, is an electrochemical process triggered by impulses carried to the axon hillock after other impulses are received on the dendrites or the cell body itself. A nerve impulse is conducted along an axon, much as a flame travels along the fuse of firecracker. This is the electrochemical process which involves the generation of an action potential, which is a wave of membrane depolarization. This wave is initiated at the initial segment of the myelinated axon. Its membrane contains a large number of voltage-gated sodium and potassium channels. And in response to a stimulus, these voltage-gated sodium channels in the initial segment, they are open and causing an influx of sodium into the cytoplasm of an axon. This influx of sodium briefly reverses or depolarizes the negative resting membrane potential, which is minus 65 to minus 70 millivolts, to positive, about 35 millivolts. And after this depolarization, the voltage-gated sodium channels close and voltage-gated potassium channels open. That causes potassium to rapidly exit the axon, and it returns the membrane to its resting potential. So the depolarization of one part of the membrane sends electrical current to neighboring portions of unstimulated membrane, which is still positively charged. So the entire process takes less than 1/1000th of a second. After a very brief refractory period, a neuron can repeat the process of generating an action potential once again. But there is a very short moment called hyperpolarization, which is caused the exits of potassium channels out of the axoplasma, and this causes the membrane potential decrease to minus 70 or more than minus 70 millivolts. This is the inhibition causing the refractory period in which the membrane cannot be stimulated to create another depolarization or another action potential. Impulse is this propagation of membrane potential changes. It means it's a wave, continues along the axolemma. And as I said, this electric impulse cannot be conducted in parts which are myelinated. That means that this impulse has to jump from one node of Ranvier to another. So the rapid conduction of the action potential is attributable to the nodes of Ranvier. Here you can see examples of voltage-gated ion channels in different moments during the generation of action potential. So the opening of sodium channels, and then like wave closing of sodium channels. So that's the wave which is spreading along the axolemma or axon plasma membrane. Myelinated axons, as I said, conduct impulses more rapidly than unmyelinated axons. This process is called the saltatory conduction. In myelinated nerves, the myelin sheath around the nerve does not conduct an electric current and forms as an isolating layer around the axon. According to the conduction velocity, we can distinguish three types of nerve fibers. First are myelinated A fibers, which are the most, which are like the fastest conduction in a human body. They are intra and extraphyseal muscle fibers. They innervate intra and extraphyseal muscle fibers. They ensure proprioception and so on. So they are ensuring a rapid reaction for some kind of stimuli. They are further dividing into 4 groups: A alpha, A beta, gamma, and delta fibers, which differ in their velocity. Then the B fibers, which are also myelinated, they represent, for example, the preganglionic nerve fibers. And the slowest are the C fibers, which are unmyelinated, and they form the postganglionic nerve fibers. They are the slowest. You can see that the velocity is about half meter per second. So that's the division of nerve fibers according to the conduction velocity. So this is a brief part about the methodical neurohistology. How can you stain the nerve tissue? Pretty important staining is according to Nissl, which can exhibit Nissl bodies and nucleus, of course. It's basic stain, but I'm not sure if you can use it normally. Very important in your clinical practice is myelin sheet staining. Most commonly, it's Luxol fast blue, LFB, which is very important if you have suspicion for demyelinating diseases, for example, multiple sclerosis and so on. Another staining is silver impregnation, according to Golgi, which outlines dendrites and axons, more the processes of neural cells. And then I think the most important today is the immunohistochemistry, because today we use special antibodies for different molecules in the nerve tissue. It's the most usable thing you can use today, I think. So here you can see the fluorescent staining and so on. So these are different types of staining. This is mentioned Luxol fast blue staining. It's very important, and it's also combined with the Nissl the Cresel violet I've mentioned before, to get the Nissl bodies visible. So these are the fluorescent markers to highlight especially the perikaryon and some dendrites, of course. So this is using fluorescein with some kind of antibody. And this is immunohistochemical staining with antibodies against some kind of proteins contained within the perikaryon of nerve cell. So those are stainings. Now let's focus on synapses. Synapses, they do have 3 main parts, as I mentioned before. First is the presynaptic membrane, which is like the ending. Usually, it's the axon ending of first neuron. So that's, for example, the bouton terminal of some axon. The cytoplasmic membrane of the presynaptic knob contains the secretory vesicles containing the neurotransmitters, for example, acetylcholine or GABA or noradrenaline and so on. So these secretory synaptic vesicles contain neurotransmitters which are necessary, they are essential for conduction of electrical impulse from one neuron to another. Between the presynaptic membrane and postsynaptic membrane is the cytoplasmic membrane of the final cell. It can be another neuron, or it can be an effectory organ, for example, striated muscle or any other cell in a body which is innervated. So this cytoplasmic membrane of postsynaptic neuron, for example, contains receptors for neurotransmitters, which are here in the secretory vesicles of presynaptic membrane. So, and between these two membranes is a synaptic cleft, which is about 20 to 30 nanometers wide. And these three parts represent synapses, like that. This is terminal button of axon. This is postsynaptic cell. It doesn't matter now if this another neuron or effectory cell. Doesn't matter now. This is cytoplasmic membrane of presynaptic neuron. This is postsynaptic membrane, and this part between the intercellular space is synaptic cleft. You can see that cytoplasm of presynaptic neuron contains lot of secretory or synaptic vesicles containing neurotransmitters. The postsynaptic cell and postsynaptic membrane contains the receptors for these neurotransmitters. Here you can see the high magnification electrogram of typical synapse in the brain. You can see the presynaptic terminal, axonal terminal, with a lot of vesicles containing neurotransmitters. And this is a postsynaptic membrane. Here is the mitochondria. And you can see very electron dense zones here, which is like accumulation of receptors for these neurotransmitters. This is a typical chemical synapses. Chemical because it uses chemical reaction between neurotransmitter and its receptor to conduct and transmit the signal. Along the axon, the signal is conducted as an electric impulse. But here, in the site of synapse, the signal has to be transformed into the chemical reaction. And that means that typical synapse is the chemical. Yeah? It has several phases. First, the action potential, which reaches the axonal ending of presynaptic neuron, opens voltage-gated calcium channels and causes the influx of calcium into the axonal ending called terminal button. This causes the exocytosis of synaptic vesicles containing neurotransmitters into the synaptic cleft. Then, of course, the calcium ions are quickly inactivated and pumped back into the extracellular space. Uh, Neurotransmitters, they diffuse into the synaptic cleft to target cell, for example, to postsynaptic membrane of another neuron or effectory cell, where they react with receptors. It causes the change of postsynaptic membrane permeability for sodium and potassium, and sometimes for another ions, but typically for sodium and potassium, which causes the inhibition or excitation of postsynaptic membrane and causes accumulation, summation of stimuli on the postsynaptic membrane. Sometimes, if there occur enough positive stimuli, for example 200, it causes generation of another action potential in an initial segment of postsynaptic neuron, and the signal, the impulse, is then spreading along the axon again, and that's the wave of spreading an electric activity via the neurons themselves. So the electric impulse, which is conducted by axon, has to be transformed to chemical reaction on the synapses and then transformed back again to electric impulse. This is the presynaptic terminal, so the axon terminal, axon ending, called, for example, it's the terminal button. It doesn't have to be, of course, it can be button in passing, for example. It doesn't matter, but that's some kind of axon ending with a lot of secretory vesicles containing neurotransmitters. Yeah. Action potential, which reaches the axonal ending, changes changes and causes the opening of calcium channels, and the calcium influx into the axoplasm and causes the exocytosis of synaptic vesicles with releasing of neurotransmitters into the synaptic cleft. Neurotransmitters then reach the postsynaptic membrane where they react with receptors and promote the postsynaptic membrane depolarization. Okay. On the other side, electrical synapses is something which is not typical in humans, but in humans we have something which is very similar to that, and those are gap junctions. You already know gap junctions, that's communication channel between neighborhood cells. They are very important, for example, in epithelium, but also in, for example, cardiac muscle, so on. In electrical synapses has some advantages. For example, there is no interruption and no delay. The chemical synapses has still some delay, time delay, because the electric impulse has to change and transform into the chemical reaction. So that's there is a slight time delay, and electrical synapse lacks this delay. And of course, signal can be spread in both directions. Some of these very similar electrical synapses, but it's not electric synapses itself, but it's very similar to that, are found in cerebellum and retina. Synapses can be also classified according to their localization. It's more logical than anything more. synapses can occur between axon and dendrite. That's the typical synapses. And then it's called the axodendritic synapse. Then it can be between axonal ending of presynaptic neuron and soma, or the body of postsynaptic neuron. It's called the axosomatic. Or it can be axonic. It means that presynaptic axon terminates in along the axon of postsynaptic neuron. There are many other type of synapses which are not so important. They occur in a CNS, but they are not so important for us. So there are some examples in varieties of synapses in the central nervous system. These are simple axodendritic or axosomatic synapses, but they also can be axonic, like that, yeah? Or sometimes they can be dendrodendritic synapses, and so on. They can be combined, not just simple, but they can be combined. That means that they terminate on the axon, or sometimes on axon and on the soma or dendrite. So the glia are non-conducting cells which are located close to the neurons. They are about 10 times more numerous than neurons. But usually they are smaller, but that's not the rule. It's just generally they are smaller. They cooperate with neurons. They ensure their nutrition. They ensure the phagocytosis, regulation of the internal fluid environment, repair of neuronal injury and damage. Of course, they ensure an isolation for nerve cell bodies and process and facilitates the rapid transmission of nerve impulses. They represent a physical support and protection for neurons. They can ensure clearance of neurotransmitters from synaptic clefts. They ensure metabolic exchange between the neurons of the nervous system and the vascular system, environment. And they can modulate conduction and transmission of impulses on the chemical synapse. We can distinguish 4 types of central neuroglia. So those are the glia present in the central nervous system. In PNS, in peripheral nervous system, there are different glia. So in the CNS, there are 4 types. First, astrocytes, then oligodendrocytes, microglia, and ependymal cells. Astrocytes are morphologically very heterogeneous cells, group of cells, and they provide physical and metabolic support for neurons in the CNS. Oligodendrocytes are special cells, smaller than astrocytes, and they ensure a formation and maintenance of myelin sheath. Microglia are the smallest cells, very small, dark, and elongated nuclei, and they possess phagocytotic properties. Last are ependymal cells, which are cuboidal to columnar cells that line the ventricles of the brain and the central canal of the spinal cord. The difference between glia and neurons is that glia can divide after birth. So their number after birth is not final. We can divide them into 2 groups: microglia, which are just microglia, and macroglia. It refers to their these three types are larger than microglia, so microglia are the smallest. Let's see this image is just the schematic topography of the brain. It's just for example, you can see neuron with axon and all four types of glia nearby. So here are the largest glia, astrocytes. Astrocytes provide mechanical support. They create some kind of new network. They have numerous processes. With these processes, they reach neurons, axons, they reach pia mater, they reach capillaries passing nearby, and so on. So these are astrocytes. Then microglia. Microglia with elongated nuclei, they can move, they are movable, and they ensure phagocytosis. Then oligodendrocyte. Oligodendrocyte produces myelin sheet in CNS. These myelin internodia enwrap axons passing nearby like that. Please note that one oligodendrocyte can enwrap more than one axon. And the last, ependymal cells. This is ventricle, for example, I don't know, third, fourth ventricle of brain, and you can see closely attached ependymal cells to create lining resembling epithelial layer, but it lacks basal lamina, so that's not typical epithelium, it's epithelioid arrangement. So four types generally: astrocytes, oligodendrocytes, microglia, and ependymal cells. You can see microglia on an electrogram and high magnification. This is the nuclei, and you can see that cytoplasm is full of lysosomes, and they have pretty much lipofuscin in the cytoplasm, some kind of inclusion. First, let's focus on astrocytes. They are the largest neuroglial cells, and they have numerous processes. They are closely associated with neurons to support and modulate their activities, and they communicate with them with large processes. So they stretch their processes from neurons to blood vessels, and also the process endings are called feet. These feet cover large areas of the outer surface of the vessel or axonema. Within the astrocyte cytoplasm, they contain intermediate filaments, which are typical for astrocytes, and these filaments contain glial fibrillary acidic protein, GFAP. This is very important because we can use antibodies to GFAP for to identify astrocytes by a specific staining. So the astrocytes can be identified microscopically by their GFAP specificity. That's pretty important. Astrocytes are connected together via gap junctions, and they form their processes which can enwrap vessels and form specific limiting membrane called membrana limitans gliae superficialis. This is like a layer helping to maintain the tight junctions of the capillaries, forming the blood-brain barrier. It's very important. You can see the image here. This is an intracerebral capillary. So that the barrier is created largely by the elaborate tight junctions between the endothelial cells, which form continuous type capillaries. And then on the outer surface, there are endings of the astrocyte processes, food processes, which maintain and fasten the tight junctions and the barrier created by endothelium. So the blood-brain barrier is made by 3 layers. First, endothelium connected with the tight junctions, then basal lamina of endothelium, and then a layer of food processes of astrocytes, like that. The blood-brain barrier restricts passage of certain ions and substances from the bloodstream to tissues of the CNS. That's very important. Substances with a molecular weight greater than 500 daltons generally do not cross the blood-brain barrier. So it's very important because it protects the CNS from fluctuating levels of electrolytes, hormones, and some kind of tissue metabolites circulating in the blood. So that's very, very important. We can distinguish 2 types of astrocytes. First are the protoplasmic astrocytes, which are more numerous in the gray matter, and they have numerous short processes which are branching. And the other type are the fibrillary astrocytes, which have longer processes with less branching, and they are predominantly in white matter. So 2 types, but you cannot distinguish them in the normal histological slide. So this is how they look like. These are astrocytes with their processes. This is the typical immunohistochemical staining with using an antibody to GFAP, and in another step using a Texas red to lighten astrocytes and their processes. So you can see rounded nuclei, a lot of processes. This is the electrogram of 2 neighboring astrocytes. So this is those are like adjacent membranes between these 2 astrocytes. You can see it's very dense, the junction between the membranes. This dense part is caused by high concentration of junctions between these 2 astrocytes. In the cytoplasm of astrocyte, you can see numerous filaments. Most are glial filaments, but you can also find their actin filaments as well as microtubules. And of course, there are some storage particles scattered in the cytoplasm and so on, and rough endoplasmatic reticulum. So those were astrocytes. Now second type, oligodendrocytes. They are smaller than astrocytes, and they are responsible for forming myelin sheets in CNS. In PNS, there are no oligodendrocytes, but the peripheral myelin is created or is formed by Schwann cells. So oligodendrocytes are analogical to Schwann cells in peripheral nervous system. So now let's talk about oligodendrocytes, because we are talking about the central glia, and then we will talk about Schwann cells and how do they differ from oligodendrocytes. So let's start with the oligodendrocytes. Very important difference between Schwann cells and oligodendrocytes is that oligodendrocytes can enwrap more axons at once. On the other hand, Schwann cells, each Schwann cell can enwrap only one axon. That's pretty important difference between these two. The myelin sheath of axon is made by concentric layers of oligodendrocyte plasma membrane. So each oligodendrocyte gives off several tongue-like process that find their way to the axons, and these processes, they wrap themselves around the portion of an axon to form internodal segment of myelin, the internodium I mentioned before. So the multiple processes of a single oligodendrocyte may myelinate one axon or several nearby axons, unlike the Schwann cells. So there are several other important differences between myelin sheets in the CNS and those in the PNS. Especially oligodendrocytes, they use different myelin-specific proteins during myelination. So the molecular expression is different between the central and peripheral nervous system, and it's very different because some of the autoimmune demyelinating diseases are focused on the CNS myelin, and some of them are focused and they are damaging the PNS myelin. So there is a difference, and this difference and this specificity of autoimmune disease, demyelinating, is caused by different specific proteins contained in the CNS and PNS myelin. So the parts which are myelinated are called the internodia, and as I mentioned, junctions between internodia are called the nodes of Ranvier, which ensure and make saltatory conduction. Here you can see oligodendrocytes with a lot of processes enwrapping nearby passing axons. This is electrongram of an oligodendrocyte. They have rounded nuclei, and sometimes the cytoplasm contains passing axons, which can be myelinated, or sometimes they are unmyelinated. And they only pass through the cytoplasm of oligodendrocyte. It can happen, but even the axons are passing through the cytoplasm. They don't have to be myelinated because they are not enwrapped by a lot of layers of myelin. This is Schwann cell. This is oligodendrocyte with a lot of process enwrapping an axon, and you can see that there are many layers of oligodendrocyte cell membrane which are encircling and enwrapping the axon of a myelinated neuron. Okay, and this is node of Ranvier because that's the junction between the internodium, this is internodium, and this is node of Ranvier. This is the difference between blue oligodendrocyte and pink Schwann cell. These cases, this and this are the myelinated axons. Myelinated axons are surrounded by a wrapping of cell membrane of an oligodendrocyte or Schwann cell. With that difference that Schwann cell can enwrap only one axon, like that. Note that oligodendrocyte can enwrap more than one axon at once. This is a case of unmyelinated axons in the PNS, in the peripheral nervous system. Even unmyelinated axons are passing through the cytoplasm of Schwann cell, but they are not enwrapped by layers. So note the difference between unmyelinated and myelinated. In the central nervous system, unmyelinated axons can pass through the cytoplasmic membrane, as in this case, but also they can be left unprotected, so they can pass completely outside the oligodendrocyte. This is high magnification of myelinated axon in the PNS, so that's the axon, that's the cytoplasm of axon, neural axon, and this is the layer of myelin sheath. You can see single layers of which represent one enwrapping of the cell membrane of a Schwann cell. So these layers are creating the myelin sheath itself. So that was oligodendrocyte, and the next one, microglia. Those are the smallest neuroglia. They normally account about 5% of all glial cells in the CNS. They are considered to be the part of mononuclear phagocytotic system, and they originate from monocyte progenitor or granulocyte monocyte progenitor. They have a small elongated nuclei. They are elongated unlike the other cells. Other cells like astrocytes, oligodendrocytes, they have round nuclei. And their processes are branching, they are spiny. They have a lot of spiny processes on their surface. They can migrate, so they can move within the CNS, and they possess a phagocytotic activity connected with antigen presenting capability. So they are antigen presenting cells. Here you can see the difference between the shape of nuclei of microglia. So the shape is elongated, unlike the other cell. Astrocytes, they have bigger, larger nuclei than oligodendrocytes. Nuclei of oligodendrocytes is usually regularly rounded like that. And those are astrocytes. So this is again the differentiation between microglia and oligodendrocytes. Please note the shape of nuclei. Schematic arrangement of the CNS again. So we have neurons with their dendrites and axon. Again, this is astrocyte. Start with that. Astrocyte, you can see pretty rounded, irregular rounded nuclei, and a cell with a lot of processes. These processes reach nearby axons, nearby neurons, dendrites, soma, and axons also. And they also reach nearby passing capillaries to create that limiting superficial membrane participating on the blood-brain barrier formation. So that's the astrocyte. Then oligodendrocytes. These are oligodendrocytes with regularly rounded nuclei, and their numerous processes enwrapping axons to create myelin sheath. Third are microglia. You can see the smallest glia in the CNS. They can migrate. They can move where to the sites where they need it, and they phagocyte, for example, the rests of damaged neurons or damaged astrocytes and so on. So they are very important for the protection of nerve tissue. And last are ependymal cells. You can see ventricle of brain with CSF, cerebrospinal fluid, and ependymal cells which are lining the ventricle. Will stay there. Ependyma they resemble epithelium. That means it has epithelioid form. The difference is that ependymal cells, they lack basal lamina. They don't have it, unlike the epithelium, proper epithelium. But these cells are still very tightly attached together to create a layer lining the ventricle. So they line the cavities of CNS, like brain ventricles and central canal and spinal cord and so on. They are connected with gap junctions and tight junctions. And on the apical surface, some of them can have cilia, maybe for facilitating of CSF flow, but we're not sure of that. So this is a central canal of the spinal cord. You can see the layer. It's discontinuous because it's already damaged by the technique, fixating technique, and so on. Inside of the central canal, there is lumen with CSF. Here, there is a layer of ependymal cells, and these cells are sometimes scattered in nests nearby in the tissue. That's pretty normal in adult human. So ependymal cells, this is higher magnification. You can see cilia, so ciliary apparatus on the apical surface of some ependymal cells. The shape of these cells is cuboidal to columnar. It depends on their activity and on the location. And these are kinocilia, so the ciliary apparatus on the apical surface of ependymal cells in higher magnification of scanning electron micrograph. Again, histological slide of the ventricle with CSF and ependymal lining with ciliary apparatus on the apical surface. Some of the ependymal cells can have also long process on their basal surface, so not the apical but the basal surface. These long processes reaches deeply to the nerve tissue, and there they are in contact with the vascular pedicles of astrocytes, or sometimes they are ending on neurons themselves. And we are not sure about purpose of these cells. Maybe they some kind of monitor the chemical composition of blood plasma, and they inform neurons about I don't know, the concentration of ions within the blood plasma and also CNF. We are not sure about that. But those tanycytes, these cells are called tanycytes, with these long processes on the basal surface, are present not usually, but most commonly on the base of the third ventricle of brain. So let's focus on the peripheral glial cells. We've already mentioned that the Schwann cells resemble oligodendrocytes in CNS, but they have more elongated nucleus than oligodendrocytes, which have more like regular rounded nuclei. The main difference is that each Schwann cell is responsible for myelination of single axon, unlike the oligodendrocytes. The myelin sheath isolates the axon, the surrounding extracellular compartment of endoneurium. And the presence of myelin sheath ensures the rapid conduction of nerve impulses, as we mentioned before. So the difference between PNS and CNS is in the structure of myelin, in the contain of specific myelin proteins, and also in the terminology, because the bundles of nerve fibers in CNS are called tracts, which are covered and formed by oligodendrocytes. In PNS, we call them nerves. So Schwann cells enwrap axons, and their bundles are called nerves, not tracts. We've already mentioned that we have unmyelinated and myelinated axons, and that the myelinated are enwrapped by layers of cytoplasmic membrane and small parts of, or amounts of cytoplasm of oligodendrocytes or Schwann cell. And unmyelinated axons are either left unprotected alone in minority of cases, and especially in the CNS, but in PNS they are usually encircled by cytoplasm of Schwann cell. So they are not left unprotected, they pass through the cytoplasm of Schwann cell. But they are not enwrapped by those layers of myelin sheath. So when we were talking about the difference between the central and peripheral nervous myelin sheath, we've mentioned those specific myelin proteins which are specific for central and peripheral myelin, and that's very important because of the specificity and targeting of some autoimmune demyelinating diseases. And the junction, the borderline between the central and peripheral myelin, is called the Obersteiner-Redlich zone. This site is usually a target where the autoimmune demyelinating process starts. For example, the multiple sclerosis first attacks this part of nervous system. So that's just it's not very important, but sometimes it's good to know the Obersteiner-Redlich zone, that the junction between central and peripheral myelin. So unmyelinated fibers in CNS freely among neurons and glial cells, but they can also be enveloped within the cytoplasm of oligodendrocytes. But in PNS, they are always enveloped within the simple folds of Schwann cells. So they pass through the cytoplasm of Schwann cells. They are not left uncovered. Already we know that unmyelinated fibers, they lack myelin sheath. That means they have no nodes of Ranvier, and that's the reason why the conduction velocity is much slower than in case of myelinated axons. On the other hand, myelinated fibers are much faster in conduction, and that's caused by that saltatory conduction we've mentioned and we explained already. Myelination is pretty difficult process which starts when the axon initially lies in a groove on the surface of the Schwann cell. The Schwann cell surface then starts to be polarized into two functionally distinct membrane domains. Between these two domains, axon immerses into the groove of Schwann cell. And these two, maybe it's better to explain here with the image. So this is oligodendrocyte or Schwann cell. Maybe it's better to explain that on a case of Schwann cell. So this is a groove in cytoplasm of a Schwann cell. This is an axon which lies there, and you can see that two parts of the Schwann cell surface become polarized and starts to encircle the axon. Here you can see that the whole axon is embedded or immersed within the cytoplasm of a Schwann cell. And those two domains of Schwann cell, you can see here, are completely encircling the axon. Here you can see this part is double membrane part that connects the two domains together and encloses the narrow extracellular space, which is here. This domain is called the mesoaxon. So mesaxon is a double membrane layer which encloses the axon within this fold of Schwann cell cytoplasm. These two domains are called the abaxonal and adaxonal domains, and this part is called the mesaxon. The myelin sheath itself develops from compacted layers of Schwann cell mesaxon wrapped concentrically around the axon. So the myelination process then proceeds by circling the mesaxon around the axon itself. And that is how one of domains, the abaxonal and adaxonal domain, one of them stays closely attached to the axon, and one of them is exposed to the external environment like that. So that's the process which proceeds as a multiple spiraling motion, which gives rise to lamellae, which are compactly arranged, and they partially fused together so like that, to create like inner and outer mesaxon. So this is inner like that and outer mesaxon. Finally, this is like final image of myelinated axon and myelin sheath. Again, that's the same image actually. And this is a higher magnification of a portion of Schwann cell with the nuclei and axon, which is enwrapped by the myelin sheath. And those are the layers. You can see here the lamellae, the layers of myelin. Okay. You already know nodes of Ranvier. Those are the gaps, the interruptions between myelinated segments called the internodium. And there is another name you should remember, and that's Schmidt-Lantermann clefts. Those are like rests of cytoplasm of Schwann cell, and these rests are trapped during the myelination process inside the layers or between the layers of myelin sheath, and they create like a lighter bands in myelin sheath. So you can see those are the yellow layers are layers of cytoplasm membrane of Schwann cell, and these lighter parts are rests of cytoplasm entrapped within the myelin sheath, and they can create lighter bands called Schmidt-Lantermann clefts. Again, Schwann cell with the nuclei and axon with myelin sheath. Pretty nice. What about the composition of myelin? We've mentioned that main composition, main structure are the lipids. They present about 70%, mainly phospholipids, but also glycolipids and cholesterol, very essential. And 30% are proteins. We already mentioned the significant differences between the CNS and PNS myelin, and that's caused by the contain of specific myelin proteins. Again, the difference between the myelinated and unmyelinated axon. This is a nuclei of a Schwann cell, so these are unmyelinated axons. Okay, like that. So note the difference. These axons, they don't have the myelin sheath, the dark layer. So Schwann cells were the first type of peripheral neuroglia we can find in PNS, and the second type of peripheral glia are the satellite cells. Satellite cells are something like they are very similar to astrocyte in CNS, but they are small cuboidal cells which surround the perikarya of neurons in the PNS. At the beginning of this presentation, we've mentioned that the accumulation or collections of cell neural bodies in the PNS are called ganglia. And these accumulations of neuronal bodies are surrounded by small, mainly cuboidal cells called satellite cells. So where can you find ganglia? Ganglia are scattered in specific locations in a human body. We will talk about it later in nervous system, but satellite cells, they can be found on the periphery of these ganglia, and they ensure microenvironmental control and nutrition, mechanical support. They isolate, they ensure the electrical isolation, and of course metabolic exchanges. So they assure the nutrition and also the degradation of waste products. This is how the ganglionic cell looks like, and these are nuclei of satellite cells. They are scattered around the ganglia cell within the ganglion. Now let's talk about the last part, the response of neurons to injury. Neuronal injury induces a complex sequence of events termed as axonal degeneration and neural regenerations. In contrast to the PNS, in which injured axons can rapidly regenerate if they have a proper environment, axons severed in the CNS usually cannot regenerate. The perikaryon, as the metabolic center of cell, must be saved for regeneration of processes, dendrites and axons. That's essential. If the axon is damaged in peripheral nerve, the distal part, that means distally from the injured part, there occurs a complete degeneration called the anterograde Wallerian degeneration. So about in the first sign of injury, axonal injury, occurs about 8 to 12 hours after the damaging. The first sign is the axonal swelling, followed by disintegration of axon. That leads to breakdown of the axonal cytoskeleton components, and these are like disassembled, like causing the disintegration of the cytoskeleton of the axon. And of course, that's accompanied by the breakdown of the myelin sheath. The most important cells in clearing myelin debris, and of course the debris from the damaged axon as well, are the monocyte-derived macrophages. The clearing of myelin debris and the rest of axon after its breakdown is essential for regeneration. So the macrophages can be activated. There are some resident macrophages in PNS which can be activated after nerve injury, and then they migrate to the site of the nerve injury, proliferate, and then phagocytize the myelin debris. In the CNS, the inefficient clearance of myelin debris due to limited access of monocyte-derived macrophages, the inefficient phagocytic activity of microglia, and the formation of the astrocyte scar severely slow and restrict nerve regeneration. So that's the difference between the regeneration of central and peripheral nervous system. So we've mentioned that the distal part of injured axon degenerate completely, but also small proximal part, proximal from the injury, degenerates. That's called the traumatic degeneration. So this complete degeneration of distal part is called anterograde Wallerian degeneration, and this partial proximal part degeneration is called the traumatic degeneration. This usually extends for only one or a few internodal segments. But sometimes, when the damage is too big, it can also extend more proximally, and it may result in death of the cell body. But that's this case if the neuron is connected only to a degenerating neuron. That means that it has one connection with this dying neuron, it dies as well. That's called a transneuronal degeneration. That means if the neuron is not connected with other viable neuron which is still alive, it will die as well as the injured neuron. So that's the degeneration. The regeneration of nerve tissue, the neural regeneration, is much more efficient in PNS from the reason I've mentioned, that the key, the essential thing for regeneration is the clearing of axon and myelin debris from the site of nerve injury. And that's, of course, that's more available in PNS than in CNS, because the CNS has the blood-brain barrier, which is not permeable, and this barrier is disrupted only at the site of injury and not along entire length of the injured axon. So that's the problem. When the Wallerian degeneration occurs and there is efficient clearing of the debris, then Schwann cells proliferate, and they divide and develop like a cellular bands that bridge a newly formed scar, and they can promote direct growth of new nerve processes. The proximal part of axon sprout new processes, which are called neurids or sprouts, and they are like nerve processes of regenerating axons. And these sprouts begin to grow from the proximal stump. They penetrate the Schwann cell bands. They are called bands of Wagner, it's not important. And they penetrate through this Schwann cell bands toward the effectory cell, for example, another neuron, or it can be striated muscle like effectory organ or something. In the case that these sprouts reach the effectory cell, in that case, usually the function is re-established. In the case that sprouts do not touch and they do not reach the effectory cell properly, the degeneration occurs and the axonal rest will degenerate, which will result in chromatolysis and maybe death of perikaryon itself. So that's the image. This is the site of the injury, axonal injury. So this is distal part and that's the proximal part. You can see distal portion of nerve fiber which degenerates in a process called anterograde Wallerian degeneration. There is also a small part of proximal degeneration called the traumatic degeneration. That's not on this picture, but it's there. Then you can see that the debris, is cleared by macrophages, and oligodendrocytes, which are dividing, and they are like undifferentiated. They create some kind of longitudinal bands through which new sprouts are growing. These are sprouts of proximal end of injured nerve, and they are growing through these Schwann cell bands toward the effectory organ, for example, the striated muscle here. So there is axonal growth toward the effectory organ, and formation of new myelin sheath from these Schwann cell bands. When the connection is reestablished, the function usually returns. So that's the regeneration. Please note that in this case, the neuronal growth has the proper environment, and that's not always present in axonal injury. For example, massive axonal injury during some kind of spinal injury, transversal sections, and so on. In those cases, the regeneration is practically very, very, very low. So full reestablishment of function is not always there. It's not present. What about those dividing cells in CNS? Generally accepted theory was that nerve cells in CNS, they do not divide, they do not proliferate after birth. But that's not completely true, because there are sites in CNS where the neural stem cells were found, or maybe that's not a good term, because neural stem cells, we're not sure if they are stem cells, but they are neurons capable of proliferation and dividing. They are present in olfactory bulb and, for example, gyrus dentatus and so on. And there is a specific expression of intermediate filament protein called nestin, which is used to identify these cells by histochemical methods. And I've already mentioned the difference between central and peripheral myelin. Those are just specific proteins which are typical for CNS myelin and PNS myelin. You don't have to remember those proteins, but maybe you could read something about the myelin basic protein, MBP, because that's the target of autoimmune demyelinating neurodegenerative disease that's called the multiple sclerosis. Okay, I think that's all. Thank you all for your attention. And in case you have some question, I'm available on this email address, so you can send me a message, and we can talk about your questions later. Thank you very much again, and hopefully you will have a nice day, and hopefully we will see each other personally soon. So bye.